Hi there, welcome to this video. So I'm going to do about adaptive immunity in this session. So I'm going to start off first of all with what is the difference between innate immunity and adaptive immunity. There's a separate video all about innate immunity, but it's important that we understand the difference between the two. So we've got innate immunity and we've got adaptive immunity. Now they do have a few different names, which makes it a bit confusing. So let's go through what names they've got. So innate immunity is the type of immunity that's not specific. So we call it non-specific. And what that means is that your body will respond in exactly the same way to any kind of pathogen. Responds in the same way to all pathogens. So by all pathogens, what I mean is a pathogen that could have antigens of that shape, a pathogen that could have antigens of that shape, an antigen that could have pathogens of that shape. At this stage, in this type of immunity, your immune system isn't differentiating between these different antigens on the surface, so it just treats them all the same, and that's things like the skin, the mucus, the lysozyme in your tears, things like that. They're there just to kill everything, anything and everything. You'll get swelling or inflammation in response to any kind of pathogen. So it's not specific at all. A really important characteristic of it is that it works immediately. And the adaptive immunity, which we're going to do today, takes time. And that's really one of the key things. So firstly, innate is non-specific. Adaptive is obviously the opposite, so it's specific. Okay, but because it's specific, it takes time for it to action, and therefore you won't be able to kill the pathogen right away. So whatever you're infected with, you're always going to start with your immune system, that's your innate immunity first of all, and then your adaptive immunity will kick in a little bit later on when it's had time to develop. There's one more word for adaptive immunity, which is acquired. Okay, it's your acquired immunity because you acquire it. Basically, you've already got some of these white blood cells in your body, but then you make more of them, you acquire more of them in response to being infected. Okay, so today we're going through all about adaptive immunity, the specific immune response. Okay, now in basics, there's two types of cells involved. So the two types of cells that we're going to look at are called B cells and T cells. Now, both of these types of cells are types of lymphocyte. So the full names would be B lymphocytes and T lymphocytes. And we've got lots of lymphocytes in our lymph liquid, which is in our lymph vessels and in our lymph nodes, okay, hence the word lymphocyte. So we've got two types of these, B cells and T cells. Now, all types of lymphocytes are made in the bone marrow, but the name of them is named after where they mature. So B cells mature in the same place. They mature in the bone marrow. So they were made in the bone marrow and they mature in the bone marrow. T cells, however, mature somewhere that begins with T. And this is the thymus. Now, your thymus is a little gland kind of in your throat. It almost looks like a little bow tie if you see a picture of it. Okay, and that's where your T cells are developed, and that's why they're called T cells. Now, your B cells and your T cells have very different roles in the body, and I've got a little way to remember these. So, the first one is a type of pencil. So if you've got a pencil there in front of you, have a look at your pencil. The chances are that on the end of your pencil it says HB. And that's going to be our little clue. Most pencils that we use are HB pencils. And that's just my little reminder to myself that the humoral response is carried out by B cells. So H and B. Now, the humoral response, the word humour in the body means fluid. Okay, so what this means is that the pathogen is in some kind of fluid. So let's say that's a blood vessel, that's your blood. Here's the pathogen floating around in some kind of body fluid in our body. Okay, it's our humoral response. I think that's an O there. On the other hand, our T cells, I've got a little reminder for how to remember what they do as well. What I think for this one, see if you can guess what I'm drawing. Let's do some Pictionary along the way. What do you think that is? So that's a cup of tea. And what's this? This is a slice of cake. So my reminder here is tea and cake. And that's a reminder to me to remember that our T cells are involved in our cell-mediated response. And what that means is that our pathogen isn't in the liquid part of our blood, it's actually hiding inside one of our cells. And that means it's less accessible for our immune response to get to it. It's harder for our white blood cells to actually access that pathogen because it's hiding in one of our cells, whereas our humoral response is a bit easier because the pathogens are accessible, they're just floating around in the body fluid. So first thing is to pause the video and make sure you can remember that B cells, where they mature and that they're involved in the humoral response and what humour means, and T cells that they mature in the thymus and which response they're involved in and what that means. Okay, so pause the video and make sure you can do that. So the next thing we're going to go through is B cells. So we're going to go on to how do B cells work next. So remember, first of all, which response are B cells involved in? So immediately when you get a B cells question, hopefully you're going to think pencil. What are pencils called? HB. So our humoral response involves our B cells. Okay, so how do B cells actually work? So remember, the full name for them really is B lymphocyte, just to make sure we know the right language. So first thing is, it's our humoral response. So that means that our pathogen is going to be floating around somewhere in our body fluid. So here's my pathogen just here, and it's got these little protein antigens on its surface. Now, also going around in your body fluid, you've got lots and lots of B cells. Now, your B cells, you actually have loads and loads and loads of different types because they each have different shaped receptors on their surface. And hopefully you're starting to see that these receptors are going to need to be complementary to the shape of the antigens on the surface of this pathogen here. So simply by trial and error, of all these B cells floating around in our system, eventually a B cell that's got a receptor complementary to that particular pathogen's antigen will bind. So remember that the orange bit there is called a receptor, and that little black bit sticking out from the pathogen that it's complementary to is an antigen. So the first step is that a B cell with a receptor complementary to the pathogen's antigen will bind. Now, we have to jump ahead a little bit here because T cells can actually help with this. Okay, so we could draw a T cell here as well, helping out. Okay, so step 2, we can say TH, which stands for T helper cells, help to activate the B cell. Now, once our T cell is activated, it's going to make it divide by mitosis. Now, sometimes that activation of just one type of B cell out of all the types of B cell that we've got, sometimes we call that clonal selection. And we can say it activates the B cell, just that B cell. You can see why this takes time, can't you? To a trial and error of all those different B cells to try out to see if they were complementary. So clonal selection activates the B cell and it makes it divide by mitosis. So what we're going to get is lots and lots of the B cells that have that same receptor on them. So we're going to have lots of B cells there. Whereas before we had a whole mix of B cells, we now, obviously we've still got those other ones, they haven't disappeared, but we have got loads and loads of B cells that have this specific receptor on them. Okay, so clonal selection activates the B cell to divide by mitosis. Now, as a fancy name for that, choosing the right one was clonal selection, and making more of them is called clonal expansion. Now, the fourth stage is that they differentiate into different types. So all of these B cells here that look like they're all the same, they're going to become 2 different types of B cells. And they're going to become 2 things called BM, which stands for B memory cells, and also BP, which stands for plasma cells. So let's have a look at what B memory and B plasma cells actually do. So a B memory cell will stay in your blood for a long time, years, maybe 10 years, long time. So stays in blood for years. And what it does is if you then get infected with a pathogen that's got these same antigens again, you'll already have lots of the right B memory cells, so you'll be able to respond faster the second time you're infected. So rather than having to do trial and error of all these different types, you've actually already got loads of the right type with the right receptor on, and therefore you'll respond faster the second time. So we call that a faster secondary response. It means if you're infected again, you'll respond faster. The other type of B cell that we make is a B plasma cell. Now, B plasma cells make antibodies. Now, the purpose of antibodies is that they can latch onto this pathogen and disable it. Okay, so they can go back and destroy that pathogen and stop that pathogen from causing a disease. They can do that in lots of ways. They can kind of stick loads of pathogens together, which we call coagulation, and then maybe a phagocyte will come along and engulf loads of them all at once. They can like bind to the toxins, stop the toxins, etc. So they stop the pathogen from being pathogenic, basically. They stop the pathogen from being able to hurt you anymore. Okay, so that's B cells. So make sure you've written that out. Make sure you've got that on a revision card or something, a summary of those 4 points, and then what the 2 different types of B cells actually do. So the next section of this that we're going to do is T cells. So remember, this system is all made up of B cells and T cells. So the next one is T cells. So have a little think, what type of immunity were T cells involved in? So remember, the fancy name is a T lymphocyte. And think about my little drawings. What should come to mind is a cup of tea and a piece of cake. So our reminder is tea and cake, and that reminds us that T cells are involved in cell-mediated immunity. So what that means is that the pathogen isn't in a blood vessel this time. The pathogen is actually hiding inside one of your cells. It's infected one of your cells. And that means that if a white blood cell was to come along, so if my T cell was here with a receptor on the surface, there's no way that T cell can bind straight onto these antigens, because it's hiding in the cell. The cell membrane of your cell is in the way, isn't it? So this is your infected cell, and this here is our pathogen. And remember, those little triangles on the surface are the antigens made of protein, and each type of pathogen has different antigens on its surface. So what actually happens is this infected cell here does something very clever. It gets hold of these little antigens from the pathogen and it presents them on its surface. So it kind of holds them out on its surface like this. And you can see why it does this, because now this little antigen is available for our T cell to bind onto, so our immune system can be activated. So when your infected cell is holding these antigens out on its surface, it's called an antigen presenting cell. Sometimes we might call that an APC. And that's because the pathogen is hiding inside your cell, so a T cell can't bind directly to it. So your infected cell holds these antigens out on its surface, so they're available for our T cell to bind onto. So step number one in our cell-mediated immunity is that the receptors on the T cell binds to the pathogen's antigens on a antigen or an antigen presenting cell. Now, just like with the B cells, this is going to take time because we've got lots of T cells in our body and they've got different receptors on them waiting for all kinds of possible infections. Yeah, so if we were infected with a pathogen that had an antigen that matched that, we'd activate that one. If we're infected with a pathogen that had antigens that were that shape, we'd activate that one. But you can see we're going to activate this one. So again, this is going to take time to select the right T cell, okay, because we've got to wait, trial and error, to try each one out until we find the complementary one. Okay, so the receptor on the T cell binds to the pathogen's antigens on an antigen presenting cell, and that only happens because they are complementary. Okay, so it's all about that complementary fit, and the word complementary is the language that we need to be using. So once that happens, it activates the T cell. Now, you might remember with the B cell, it had to be also activated by a T helper cell. T helper cells aren't needed to activate T cells. They can just be activated on their own. Okay, so this activates the T cell. And then it was going to do the same as before. It's going to divide by mitosis. So then we're going to have lots and lots of the T cells that have this specific receptor on them. So loads of them that have got this same triangle shape receptor. And then the same as before, they're going to differentiate into different types. So the different types of T cell that we're going to make are TM, which stands for memory. They're exactly like the B cells. They're going to stay in your blood for years, and then in secondary infection, if you're infected by the same pathogen again, you'll be able to respond faster in the secondary infection. We've got T helper cells. Now, we already know about T helper cells. They activate the B cells. Now, I'm going to add a little bit more detail now about how the T helper cells activate the B cells. So they release a chemical called a cytokine. Now, there's different types of cytokines. The one that they release is called interleukin. Okay, but it's really important that we know that T helper cells release cytokines, and it's those cytokines that help the B cells to divide, and also they stimulate phagocytases, so we're more likely to engulf those pathogens and get them out of our system. And we looked at phagocytosis when we did innate immunity, so hopefully we know about that a little bit as well. The third type of T cell is a T killer cell, TK. So that stands for a T killer cell. And the cell that it wants to kill is the infected cell. So remember, this is our cell-mediated immunity. So this is when the pathogens are inside our cells. So we want to go back and kill all those infected cells. Now, the way it does that is it basically makes holes in the membranes. And if you've got holes in a cell membrane, a cell will never survive, okay, because of osmosis, water and other ions and everything will just go in and out uncontrollably, and it will cause cell death, which we call apoptosis. All right, so it will kill the cell, hence the word killer cell. All right, so that's the basics of our specific immune response, the B cell process and the T cells process. Now, everything we've done about so far has been what we call our primary response. And what that means is this is what happens the first time you're infected. Okay, so these B cells and the T cells that we've just gone through, all of that happens the first time we're infected. So what do we mean by a secondary response? Now, the same thing happens, but this time in the first step where we're trying to find the B cell that's complementary to the pathogen or the T cell that's complementary to the pathogen, we won't have kind of an even amount of all of those different shaped receptor ones. We'll have loads of memory cells from any shape that we've been infected from before. So we will already have memory cells. So there were two types of memory cells, weren't there? There were T memory cells and there were B memory cells. So those memory cells will have stayed in our blood for years. And if we're infected a second time by the same pathogen, and this is really important that it needs to have the same antigens on the surface. Now, sometimes pathogens actually evolve. So, for example, the influenza virus, you could catch influenza this year and have a primary response and it would have those antigens, but by next year those antigens might have mutated and be a different shape. So even though you'd think, oh, I've had flu before, I should be on my secondary response now, I shouldn't get it so bad, actually in the case of the flu you would get it just as bad because they mutate so fast. But other pathogens, their antigens might stay the same for years and years and years, in which case if you've had it before, the next time you get it, you'll get better so much quicker. And sometimes you get better so fast, you destroy them in your system so quickly that you don't even realize that you had an infection. Your immune system has worked before you felt any symptoms. So there's a really famous graph about primary and secondary response that you need to know. So it's about the number of antibodies, and it looks like this. So this number up the side here is the number of antibodies, and the axis on the X down here is time. So what this graph shows is that the first time you're infected, just put a number 1, you slowly make antibodies. And the reason you do it so slowly is because you've not got many B cells that have the complementary receptor. You've got to wait for the right one to bind. You've got to wait for it to be activated. You've got to wait for it to divide by mitosis, differentiate, and then the plasma cells to make the antibodies. So you've not got many complementary B cells in your system. You've got to wait for all that process. So it happens quite slowly, and you don't make that many. Okay, so you don't make many antibodies, and you're likely to feel very poorly. Okay, because that's going to take a long time, and even in the end, you don't get that many antibodies anyway. Now, if you're infected by the same pathogen again, just here, second time you're infected, this line you can see goes up much faster. Okay, so this time you make antibodies much faster, and look how many you make, all the way up to here on the axes. Okay, so you make antibodies faster and you make many more antibodies as well. And the reason for that is because you already have lots of B memory cells in the blood from your first infection, and therefore you will respond much quicker the second time. Okay. So that's what we call our primary response. That just means primary, like your first school, your primary school, your first time you're infected with that pathogen. And then your secondary response is like your second school, secondary school. It's the second time you're infected with a pathogen. And the important thing is you make antibodies faster, you make more antibodies. And the reason is because you've already got those B memory cells in your blood. Okay, the next thing we're going to look at is different types of immunity. So sometimes we develop immunity naturally. Oops. That's what we've been talking about so far. But sometimes you could develop immunity artificially. So natural immunity develops, like we just discussed, it happens when you get infected once. So after infection, you build up some natural immunity because you make memory cells. Okay. On the other hand, artificial immunity is when the same thing happens, but it's done artificially. So this would be when you get a vaccine. So the first time your body is exposed to that pathogen with those antigens on its surface, it wasn't a natural infection, it was a vaccine. So when you have a vaccine, a dead or a weakened version of the pathogen is injected into you, and then your body goes through that same immune response. So that triggers an immune response, just like we've just learned about. Okay, and that is going to then make memory cells exactly the same. So if you're infected for real, again, in the future, you'll already have all those memory cells ready. Now, there's two other categories we can talk about as well, which are active versus passive. Now, everything we've talked about so far has been active immunity. So what we've been talking about is your body actively making its own antibodies. It's good to put the word actively in there because then it links the active bit. So passive is the opposite of that. So we can sometimes be given ready-made antibodies. So it's not our immune system making our antibodies. We're just given antibodies already made. We don't make them. Now, why would we give someone antibodies already made? There's a few benefits to it. Or there's one main benefit, I should say, really. It's really fast. Okay, it's really quick. That's the benefit of passive. Whereas active takes time. Okay, it's quite slow. Now, on the other hand, there's a reverse argument because the active immunity will last a long time. Because in active immunity, you're going to make memory cells. You'll make antibodies as well, but you'll make memory cells too. And that's why it lasts a long time. Whereas passive immunity, because your immune system was never triggered, it's only temporary. So it's kind of like a good and a bad thing. So that's a good thing about that. That's a bad thing about that. Whereas on this side, that's the good thing and that's the bad thing. So sometimes they might give you examples and ask you which categories you think that they fit into. So let's have a think. So for example, if we think about breast milk, so a baby, if they get breast milk from their mother, that contains some antibodies. So the newborn baby gets some immunity straight away when it's born, okay? It's perfectly natural, okay? And it's passive. All right, it's natural, it's not artificial, it's natural, and it's passive because the antibodies are already made. Okay. Next example could be, what about if you were given a vaccine? We've sort of talked about that already. So it's artificial, it's not a natural thing. Is it active or passive? It's active because your body still has to respond to this pathogen, even though it's not going to hurt you. Okay, so it's active. Another example could be just getting an infection, a natural infection. So if you're infected with somebody, it's definitely with the pathogen, sorry, it's definitely natural and it's active because you've got to make your own antibodies. Now, the last example that fits into the last kind of combination is when you get an injection of antibodies. And sometimes you get this for things like tetanus. So if something happened to you, or rabies as well, if tetanus, if like you got a rusty nail through your foot and you needed some immunity from it really fast, they might give you an injection. So it's artificial, but it's already got antibodies in it, so it's passive. Same thing for rabies. If you got bitten by a wild dog or something, it's artificial because they give you an injection, but it's passive. They give you ready-made antibodies because you need to be treated really quickly before it makes you ill. So sometimes you need to work out the combination. Is it natural or artificial? Is it active or passive? Right, we've got one more section to do, which is about autoimmune diseases. Okay, so autoimmune diseases are when your immune system basically goes wrong and it attacks self cells instead of non-self cells. Okay, so your immune system attacks self cells. Normally it should attack non-self cells. So your own cells have antigens on their surface as well, and it should be able to tell that a certain shape of antigen means it's one of your own cells, whereas a different shape antigen means it's a pathogen. Okay, so it can tell your cells, so self cells from non-self cells, by the type of antigen that they've got on the surface. But sometimes we get a disease, which means we've got something wrong with our body where our immune system can't tell the difference, and for some reason we start to destroy our own cells. So the first example of one of these that we're going to look at is diabetes. And we have looked at diabetes also in the controlling blood glucose lesson. So the full name of diabetes is called diabetes mellitus. Okay, and for each of these 4 diseases that we need to know the cause, the symptoms, and the treatment. Okay, so first of all, the thing that causes diabetes is that we've got some antibodies that are destroying our own cells. So the word auto means destroying your own things. So auto antibodies are antibodies that aren't destroying pathogens, they're destroying your own cells. Okay, so auto antibodies destroy your B cells, or beta cells, sorry, I should say, in the pancreas. Now, as we know from our blood glucose lesson, the beta cells make insulin. So people with this type of diabetes can't make insulin and therefore can't control their blood sugar levels. Okay. There's lots and lots of symptoms, but the most famous symptoms are thirst. So you feel thirsty a lot of the time, and also frequent urination. You go to the toilet a lot of time. Okay, so very common symptoms of diabetes, and often some of the first things that people pick up when they get diagnosed. For the treatment for this type of diabetes, you can have insulin injections because you're not making insulin. So if you inject insulin, then you can control it. Some people now also have an insulin pump fitted, which basically injects the insulin whenever they need it. So it's monitoring the insulin levels and giving them insulin when it's needed. It's monitoring the glucose level really in the blood and giving insulin when it's needed. There's 4 different diseases that we need to do for this. So the next disease we're going to do is MS. So do you know what MS stands for? It stands for multiple sclerosis. So first of all, what causes multiple sclerosis? So obviously it's your immune system attacking your own cells, and this time it's the myelin sheath of the brain and the spinal cord. Okay, so your immune system attacks the myelin sheath in the brain and the spinal cord. So you can see it's going to affect your brain and your spinal cord. So we learnt about the myelin sheath, if you remember, when we did neurons, and we said that around the axon, we've got this myelin sheath. Okay, and that helped the nerve because we did saltatory conduction, if you remember, so we could transmit the signal even faster. Now what happens is that myelin sheath gets inflamed, so it gets even bigger, and actually it pushes down on that axon, and it means that the axon can't send nerve transmissions properly anymore, so it disrupts the nerve transmission. So what, for the next pox, is the symptoms of this? So you're going to feel very tired, but also kind of coordination things. So it's hard to walk, it's hard to see, it's hard to control your limbs. Sometimes you can't even feel your limbs. Balance, coordination problems, lots and lots of problems caused by this nerve damage. Okay, so coordination, walking, bladder control. All right, really, really difficult. And the treatment for it, you can have steroid medication, first of all. But also just kind of what anybody would do if they had those kind of symptoms or uncomfortable feelings. So things like if you feel stiff, take some exercise. Also avoiding painkillers, because some painkillers make you more tired, and trying not to always use like walking aids and things, but it's better to just exercise and try and keep the muscle and keep the nerves in use. So steroids and exercise are the main things. Okay, 2 more diseases to go. So we've done diabetes and MS. The next one is Crohn's disease. Just double check your spelling of Crohn's. It's a bit of an unusual one. So Crohn's disease affects your digestive system. So your immune system this time attacks the cells in your intestines. So it's the cells that are making up your intestines now that are getting damaged. You're going to have symptoms from your digestive system. So your stomach's going to hurt, diarrhea, sometimes some blood in the feces, sometimes a lot if it's a bad episode. Feel very tired because your intestines aren't working properly, not absorbing all those nutrients, and ultimately weight loss if you're not absorbing nutrients. Okay, so diarrhea. Oh, can never spell it. Let me double check the spelling. Hang on. OEA, sorry. Scribble that out. OEA. So diarrhea, stomach pain, blood in the feces, tired. All right. So how do we treat that? Then again, steroids. Steroids are medication that stop inflammation. So again, you would have an inflamed bowel. So steroids would help. If you can't struggle to kind of get food through your intestines, sometimes liquid food is easier to manage. You can take medication to suppress your immune system, to stop your immune system from keep reacting all the time. And sometimes you actually need surgery to remove sections of your intestines if it's got too bad. Okay, so liquid food, surgery, remove parts intestines. Okay, one more to go. So the last one we're going to look at is arthritis, and the fancy name for this is rheumatoid arthritis. Checking the spelling again. Rheumatoid arthritis. Okay, so arthritis, you might know, it kind of makes your joints really stiff. They swell up. Sometimes if you've got arthritis, you might notice people have really swollen hands. So your immune system is attacking the tissue in your joints. It is painful as well, so the tissue then becomes sore and inflamed, and then it releases some chemicals that can actually cause further damage to your bones, cartilage, tendons, and ligaments in the joint as well. Okay, so we can say inflammation. Okay, and if you want to add that it can affect the other parts of the joint as well, it's up to you. So the symptoms are it hurts, stiff, swollen. Also, you would feel tired and weight loss sometimes as well, depending on whereabouts in your body it affects. So pain, swelling. What else did we say? Stiff, tired. So how do we treat arthritis? You can take tablets to reduce the effects of the chemicals released in inflammation and painkillers. You would actually take non-steroidal anti-inflammatory drugs for this one. And then you can take steroids like we talked about before, and also physiotherapy. Okay, so let's put steroids just because it's easy to remember, because then it's the same for lots of them. Painkillers. Exercises by physiotherapy just gives you exercises. Try and keep your joints moving. Okay, and that brings us to the end. That's all the diseases you need, those 4, and it's always going to be cause, symptoms, and treatment. All right, so that's the end of this video.